Hello, everybody. How are you today? Very cold. Can you hear me? Yes, good. Thank you. So this week is lab quiz week for the muscles, which is the hardest lab exam, I think.
Yes, it's definitely the hardest.
Lots to study, but it's fun for some people, right? It's important. Okay, just here's the time. Wait a few more minutes and then we'll start. The only 10% of the class is here. Incredible. You have your exam this week already? I thought this week was just the quizzes. Oh, I see. You had your quiz last week.
Professor, the final exam's on the 16th of December, right?
Correct.
Is there any way I can take it on the 17th? Because I have another exam on that day.
No, I'm sorry. We
It's not.
cannot change. You have to understand that there's close to 700 students. I cannot make changes.
Oh, because I saw the calendar and it said that makeup exam's on the 17th.
Yeah, but that's
Uh,
only if you're sick or if there's an emergency. You can ask the
oh,
other professor to change the exam.
whom?
You can maybe ask the other professor to change the date.
Oh, like my other subject. Oh, okay.
Yes, right?
Oh, I'll ask him. But he's like really rude. I don't think he's gonna listen.
So
But
we have
you're
this
really
time.
nice. That's why I asked you.
No, but remember, we have so many students. If I make exceptions, it's impossible. I have to
Mhm.
have very strict rules. It would be easier to say yes, but I can't. And that's from the first day of class, I explained that. Okay, I think it's time to start. Hmm, Okay, so today's conversation is going to be about electrophysiology. So some of you might not find it so interesting, but it is. It's the base for how we can move, we can respond to different changes in the outside environment and inside our body, how it is all this coordinated through the neurons, right? Well, we have the endocrine system also, but now we're concentrating on the nervous system. So, how does all this happen? So, the basis is the generation of an electrical current. And that electrical current is possible because of ions going in and out of the plasma membrane of the neuron. And that is highly, highly regulated because of the properties of the, the neurons. So, what you see here is the axon terminal of one neuron, which we call the presynaptic, and the blue neuron is the postsynaptic. And you see that these processes of the axon terminal are projecting towards the dendrites. This is the cell body, and these are the dendrites. And remember that there's never a physical contact. There's very close proximity, but there's always a space in between the presynaptic membrane of the axon and the postsynaptic membrane of the dendrite. And that's known as the synaptic cleft. So the contact, the continuation of the propagation of the signal from the presynaptic neuron to the postsynaptic neuron depends on the release of neurotransmitters, of chemicals, and diffuse across the synaptic cleft and bind to receptors. And that's a big, big advantage that there is this space, because it allows for different chemicals with different properties. Some are excitatory, some are inhibitory. So you can think, if you think about a car, some act as accelerators, so the signal will be propagated from the presynaptic neuron to the postsynaptic neuron. Other chemicals are inhibitors. They act as the brakes, so they prevent the propagation of the action potential. So question, so presynaptic and postsynaptic never touch, right? The synaptic cleft is always in between, correct? And the beauty of this is that the propagation of the action potential from pre to post depends on what are the neurotransmitters that are going to be released at the axon terminal, and what are the receptors for those neurotransmitters at the membrane of the postsynaptic neuron. And today you're going to realize how complex that is. We're just going to talk about channels and different types of channels and different types of neurotransmitters. Okay, so here you see, you've seen different versions of this circuit, where in this case you see the skin, and you have a stretch receptor at the periphery of the skin. And if there's pressure exerted upon that receptor, if it's strong enough, then it will initiate an action potential. It comes down the sensory neuron. Here's the cell body of the sensory neuron in the ganglion. And the action potential travels. This is a cross section of the spinal cord. Remember, the gray matter is in the middle. That's where the cell bodies are. The white matter on the outside contains the myelinated neurons or axons. So you have then the action potential going through the interneurons, so synapses with an interneuron. And the interneuron synapse is with a motor neuron that has the cell body, the gray matter of the spinal cord. So both the interneuron and the motor neuron have the cell bodies in the CNS, the central nervous system. The cell body of the sensory neuron is in the PNS, the peripheral nervous system. So the action potential travels down the motor neuron, and then it reaches the effectors. The effectors can be muscles or they can be glands. So this is basically the trajectory that that electrical current, that the action potential is going to follow to bring to the central nervous system information of changes that occur at the periphery, and then triggering a response that is going to be conducted by the motor neurons. Okay, and you've seen this actually here also, but I want to point out a detail here. So this is the spinal cord, and you see the sensory neuron here. This is not correct because the sensory neuron doesn't come out of the body, so it should go through here and then all the way out. But anyway, just to make it simple, so the sensory neuron with the cell body comes to the spinal cord. Now, here, I hope you see it, there's several interneurons. And the advantage of having these several interneurons is that the, the action potential that is initiated by one sensory neuron, let's say, can be carried, can be conducted to several interneurons, and then from several interneurons, several motor neurons can be activated. So the red motor neuron is going to activate or send a message to the biceps, which is a flexor muscle. But also the same action potential carried by the sensory neuron is going to be transmitted to another motor neuron that now is going to carry the action potential to the triceps, which is an extensor. So by one action potential can generate more than one action because of the interneurons that will conduct the action potential to different motor neurons. And here we could add even more complexity. If there would be more interneurons, they could carry the same action potential to the other side, to the contralateral side, and they could conduct up to the brain. Now, in this case, this circuit doesn't show action potential traveling to the brain, to the higher center. So we call this a reflex. So reflex is a very, very quick action potential or circuit that allows for a very quick response because it doesn't even have to go to the brain to generate a response. It just goes in and out from the spinal cord. So this is what we call a reflex.
Professor,
Yes.
I had a question. So the two different action potentials that are going to the biceps and triceps, are they coming from two different interneurons?
Yes. You see here, here's one interneuron, and here's another two. Actually, here there's two interneurons, and here there's two others. And they synapse with two motor neurons. Yes. So you can have several interneurons carrying the action potential from one sensory to several motor. Okay, so, and you saw this before, that you have the flame that is going to activate a temperature receptor, and only when the flame is hot enough will it trigger an action potential. So remember, resting membrane potential is minus 70 millivolt. So if the flame is not strong enough, the heat generated by the flame is not strong enough, and just changes the potential from minus 70 to minus 65, from minus 70 to minus 60, nothing happens. That will, we call that a local potential. It will die out. However, if the flame is strong enough, the heat generated is strong enough and reaches threshold, which is minus 55 millivolts, so it goes from minus 70 to minus 55 millivolts, then that activates that area, the trigger zone. Remember, where the cell body meets the axon, where there's a huge amount of voltage gated sodium channels. So when you reach the minus 55 threshold, then the action potential is activated, and then you have an action potential traveling through the sensory neuron, going to the spinal cord, coming down the motor neuron, and causing movement because it activates the two muscles, right? So there's several questions here. When does an action potential then generate a reflex versus going all the way to the brain? So it depends on which interneurons are present there and how the connection between the sensory neuron and the interneuron and the motor neuron. And the neuron that carries the action potential there, specific sensory neurons that will carry the action potential to the brain. And we will see this in a few more slides. You will see that. Why does it take a few seconds? It's not seconds, it's milliseconds for you to feel pain when you get burned. Okay, so the reflex is very quick. So you take off the hand much quicker than when you feel pain because the signal has to travel to the brain for you to feel the pain of the heat. But the reflex is very quick. The stimulus, exactly, Kevin, the stimulus has to be strong enough to be conducted to the brain. Okay, I can post everything. Yes. Okay, so let's just see what's happening here. So you see the flame until it reaches a certain point that it generates an action potential. It goes here, and here's the interneuron in green. And now the action potential travels down the blue motor neuron. And here you see the biceps being activated. Okay, so this is one of the quickest circuits. It's called a reflex arc. But go back here. There. Okay, so here now we're making things more complicated. Now you want to feel pain if it's strong enough, right? In the reflex, the order is sensory neuron, interneuron, motor neuron. No, 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 no. Once the action potential is going to be established here at the receptor. So it has to be strong enough to go through the sensory neuron, travel to the interneuron, and go to the motor neuron. So the action potential is what travels a long distance. The local potentials that only go from minus 70 to, let's say, minus 60, they won't travel, they will just stay there. So here you see that the sensory neuron is synapsing with these higher order neurons that bring the action potential to the brain. So you see one goes directly to the thalamus. So the thalamus is an area in the midbrain that's very important, that most of the sensory messages end up in the thalamus. And then from the thalamus, they are going to be distributed to the different areas of the brain. Here, we are considering the pain and touch. So it synapses with this neuron that goes to the primary somatosensory cortex and some aesthetic association area. So this is a third order nerve fiber. So why is it called a third order? This is the first order, then this is the second order, and here's the third order. So that means that this action potential carried by the sensory neurons involves a pathway that involves three neurons: first order, second order, and third order. When the action potential is not at rest anymore, it's minus 70, sensory neurons sensory neuron sends info to the interneuron. No. When it's at -70, that means it's resting. Nothing has happened. There's no electrical current. It's only the action potential that travels. So then you see this other pathway. You have the first order neuron, the second order neuron that conducts the message to the reticular formation, another area of the brain, and then to the hypothalamus, and then to the pain area. So you saw the reflex that goes in and out very quickly. So the circuit only goes from the periphery to the spinal cord and right out to generate a response. Or if it's strong enough, then it's carried up to the brain, and that's when you feel pain. Okay, so this is shown here a little bit. Let me see. Go here. So here's another view where you're going to see the activation of the pain receptors. So the action potential is strong enough to go through these wires that go all the way up the spinal cord, and then from the spinal cord, they're going to continue to travel up to the brain. You see here the synaptic cleft and the neurotransmitters being released. And then it goes up to the brain, to the pain center in the brain. Now, wouldn't it be nice if I could switch? Okay. So can you see the slides? Yes? Can someone tell me? Yes. Okay. So you see how all this happens. The traveling, it's indeed an electrical current that's traveling through these wires, and the wires are the neurons and the network. So many neurons can be involved and so on. So how can we measure this electrical impulse, this action potential with electrodes? These electrodes can be made out of gold or something, that a metal that's very, very sensitive. And you put one in one area of the neuron and another one in another area. And you can measure the change in potential at the membrane, at the plasma membrane. Okay, so now let's focus on the different potentials that are occurring on a neuron at the different time points. Okay, so we have the resting membrane potential. When the neuron is at rest, there's no stimulus. So nothing is happening here. Everything is quiet, but not really static. It's still dynamic because the resting membrane potential has to be maintained. And you will see how. Then if there's a very light stimulus, then you have a local potential that is generated. And these local potentials occur at the dendrites or cell bodies. So you have local potentials that go from -70, which is the resting membrane potential, to -65, -60, but never reach the threshold of -55. So these local potentials that occur at the dendrites and cell body, they travel short distances and they can change. They can go from to -65 to -60 and so on. And then you have the action potential that travels the long distance down the axon. And this is an all or none response. So it goes from -55 quickly up to +30 millivolts. And this is that threshold, the -55 millivolts, has to occur here at the trigger zone. So then it goes down the action potential. So what I described the resting, the local, and the action potential. The local potential travels very short distances, just in the cell bodies and dendrites, and the action potential travels long distance down the axon. Okay, now I've been making things very simple. It looks like there's just one synapse. But here you see a neuron, a pink neuron, and you see all these axon terminals coming from many different neurons. They look like worms with the synaptic knob at the end. So that means that the pink neuron is a postsynaptic neuron. And here, what it's showing is that it receives signal from many different presynaptic neurons. So all these are presynaptic neurons that are releasing their neurotransmitters onto the pink postsynaptic neuron. And it's the addition, the sum of all the effects of all these different presynaptic neurons. It's going to decide if that action potential, that change in potential is going to be converted into an action potential and traveling down the axon. So the local potential is communication within the cell body and the dendrites, yes, of a single neuron. Yes, perfect. Okay, so neurons are excitable cells, just like muscle fibers. The current is carried by ions, not electrons. So the local and action potential occur because of opening and closing of ion channels. So these ion channels that are on the plasma membrane, the ion channels means channels that open and close and allow sodium, potassium, calcium, chloride to get in and out. So that's why they're called ion channels. And their opening and closing is highly regulated. So a neuron has to decide which local potential it must convert into an action potential. No, it doesn't have to make the decision. The decision comes from the change in potential, right? So if you have a lot of axon terminals releasing neurotransmitter on the postsynaptic neuron, and some will be inhibitory, some will be excitatory, until the change, the sum, reaches minus 55 millivolts. The decision is to reach threshold. Once it reaches threshold, minus 55 millivolts, then it will be converted to an action potential. Right, so the local potential, the sum, let's say the sum of the local potentials has to be strong enough to reach threshold. Perfect. So the local and action potentials are initiated by ion channels that open and close in response to different stimuli. And these are located at the plasma membrane. But what maintains, so you also have the resting membrane potential that depends on a certain type of channels. So how is this possible? It's possible because of the properties of the plasma membrane. Remember, we always said that the plasma membrane is not a semi-permeable membrane. It's a selectively permeable membrane. And now the difference here is semi-permeable. That means that it would allow ions and molecules and whatever to go in and out depending on the size. But selectively permeable means it's not only the size, but also the charge of the molecules. Some can go in, some cannot go in, depending on their charge and the presence of these ion channels. So the plasma membrane is an excellent insulator because of the phospholipid bilayer. Remember, it generates a charge surface on the outside and on the inside that will repel ions. positively or negatively charged ions. And those ions can only go in through the channels. And that is very, very important for an action, for a local and action potential to be generated. So the properties of the plasma membrane are essential for these different kinds. Oh, I didn't go all the way up to there. Wait a minute. Okay, so now let's look at channels, different types of channels that allow for this uh, electrical current to go from the periphery to the brain, from the brain out to the periphery, and allow survival, right? We are totally dependent on this electrical current to survive, because without electrical current, we cannot adapt to changes in the external and internal environment. Oh, so let's see here. Okay, thanks for telling me. Okay, so here are different types of channels. Here, in this panel, we haven't talked about these channels yet. These channels are very important for the resting membrane potential. These channels are known as leakage channels. So that means that these channels are dedicated either to sodium, the orange one, or to potassium, the blue one. Of course, they're not these colors, right, on the cells. But these channels, the opening and closing of these channels is not regulated. They open and close randomly. And that's why they're called leakage, because they allow for, remember that there's lots of sodium on the outside. So these channels will allow sodium to come in a random manner. The potassium, the same thing. There's lots of potassium in the inside. So these channels allow for potassium to go out without being regulated. You don't need any stimulus. So these leakage channels are very important for maintaining the resting membrane potential. So what I'm showing here is the extracellular fluid, the plasma membrane, and the intracellular fluid. So this would be intracellular, extracellular. And here are the concentrations of potassium and sodium. So you can right away see that the concentrations of potassium is much lower on the outside than in the inside, and the sodium the opposite, high concentration on the outside and less in the inside. And how is this possible to maintain this difference in concentration of potassium and sodium? Now you switch here to the sodium potassium pump. That's why this sodium potassium pump is so important and uses 40% of the ATP, because the sodium potassium pump with the leakage channels are essential for maintaining resting membrane potential. So the sodium and potassium ions travel in opposite directions. So with the leakage channels, for example, the sodium will come in because there's more outside than inside. So this is not active transport. This is just the channel is open randomly. And because there's more sodium here, it goes in. But now the sodium potassium pump, remember, you need active transport, because what does it do? It brings sodium outside and potassium inside, so that maintain high levels of sodium on the outside and high levels of potassium in the inside. So the leakage channels and the sodium potassium pump transport the ions in opposite directions. Sodium goes in through the leakage, goes out through the pump, and potassium goes out through the leakage and comes in through the pump. So these two are very important for resting membrane potential. Here, the channels in the middle, which are gated, that means they're opening and closing is highly regulated by the different types of stimuli, of the release of neurotransmitters, or even mechanical stimuli, which you will learn today. So these channels are closed, and for them to open, you need chemicals, or you need a mechanical action, or you need a change in voltage. So these are gated ion channels, and these are involved in the local and action potential. Yes, but this here, the pump uses most of ATP. Okay, so here we have the leakage ion channels. You see here the potassium leakage channel that allows potassium to go out because there's more inside. And since it's not regulated, you just open, it's like opening a window and allowing potassium to go out because there's more inside. So they open and close randomly. This is what is important to remember here. And here's the pump, which uses ATP because there's active transport, and therefore it allows for sodium to go out against its concentration gradient and potassium to go in against its concentration gradient. So it moves from the outside, where there's less, towards the inside. So these two are very important in maintaining resting membrane potential. And this here is shown again. So that just that the ions, sodium and potassium, move in opposite direction in leakage versus the pump. Okay, and here I just want to remind you that the properties of the plasma membrane, being a selectively permeable membrane, are essential for this electrical current to be able to move, to be propagated. So here you have the change in concentration, so the chemical gradient, and here you have a change in charge, which is the electrical gradient. And these two together allow for the resting membrane potential to be minus 70 millivolts. Okay, now, which is the ion that contributes most to the resting membrane potential? So you can guess, but
Will it be a negative ion? Because it's
it's potassium. Remember, potassium is very important. Remember when I told you in some lectures, back lectures, that if you would give a patient a solution that's high in potassium, that person will die because it cannot, of cardiac arrest, because it cannot maintain the resting membrane potential. Why? Because you have so much potassium outside that then no more potassium will come out, and you don't have that differential charge, and therefore there's no resting membrane potential. Okay, so let's see, how is it that potassium is able to generate a resting membrane potential of minus 70 millivolts, right? So, so you know the differential distribution of the ions. Outside sodium and chloride, inside potassium and proteins. Well, you could die from eating too much bananas or peels, potato peels. No, we need potassium, but not in exaggeration. Okay, so here is the clue. The membrane, the plasma membrane, is much, much more permeable to potassium than to sodium. And why is that? Because there's 50 to 100 times more leakage channels on the plasma membrane for potassium than for sodium. So that means there's 50 to 100 times more leakage potassium channels on the plasma membrane than sodium. Therefore, of course, there's much more potassium leaving the cell. And if there's much more potassium leaving the cell, then it becomes negatively charged, besides, the charge contributed by proteins and so on. But here, the very, very important factor is that at the plasma membrane, there's 50 to 100 times more leakage channels for potassium than for sodium. So there's much more potassium that leaves than sodium goes in, and therefore the inside becomes negative and it reaches minus 70. So the resting membrane potential is minus 70 millivolts, and potassium is the ion that has the greatest influence. Okay,
Professor,
yes,
so when potassium leaves the cell, it goes down to -70, and that's how it establishes the resting potential, right?
yes, but I'm lying a little bit because the potassium, if we just consider potassium, then the resting membrane potential should be minus 90 millivolts and not minus 70. And that's because there's other ions that contribute. So it's the resting membrane potential in a neuron is minus 70 millivolts. Okay, but now let's see what happens to potassium. Why is it so important? Okay, so potassium has two forces acting on this ion, right? So there's a chemical gradient because there's more potassium inside. So it's pushing potassium outside. So you have that force that's pushing potassium outside through the leakage channels because there's so much inside. However, there's an electrical gradient. So that means the inside is negatively charged. So it's preventing the potassium to go out, right? So concentration is pushing it out, but the negative charge prevents potassium to go out because potassium is positively charged. So when there's an equilibrium, that's the resting membrane potential. Potassium equilibrium, the potassium equilibrium potential. It depends on these two forces, one pushing potassium out and one keeping potassium in. When there's equilibrium with those two, between those two, then that's the minus 90 millivolts. But because other ions, such as sodium, can contribute to that resting membrane potential, so that's why it's minus 70 millivolts. So this is very complex, and can you imagine figuring all this out? But remember, potassium is very important, not in huge amounts, because that can be lethal, but it's very important to maintain the resting membrane potential. And the resting membrane potential is also critical because if you don't have a resting membrane potential, then you cannot activate the neurons. So here, what I'm showing you is a section of an axon that has the leakage channels that allow potassium to leave the cell. And then you have the sodium potassium pump that brings potassium inside. And these two work together to establish the resting membrane potential. Okay, and here I'm showing the importance of potassium. So I don't know if any of you, many of you probably were not born in 1990. I was, for sure. And there was Dr. Kevorkian was in the news. He was a doctor that believed on euthanasia. And in the United States, it was not legal. So he actually went to jail. So the first person that he helped to commit euthanasia, which is assisted suicide. So if someone is not is so sick that they don't want to live anymore and they decide to die, that's what is called euthanasia or assisted suicide. And Dr. Kevorkian developed a strategy because doctors cannot kill a patient, right? Because of the oath. They cannot kill a patient. So a patient has to do it voluntarily. So Dr. Kevorkian devised a strategy for the patient voluntarily to commit euthanasia. So there's a woman with Alzheimer's, but she was still a little bit with some cognitive function, and she didn't want to go through anymore. So she approached Dr. Kevorkian. So this is what the strategy is. So an intravenous is applied to the individual, and when the individual is ready, opens the intravenous. And then what happens, the first thing that comes out is an anesthetic. So that means the patient is knocked out and has no notion of what's happening after. But this is a voluntary act. It's the patient that opens the intravenous so that the anesthetic first comes out. After the anesthetic, you have a high concentration of potassium chloride coming out and being administered to the patient. And therefore, there's no resting membrane potential. The heart cannot be stimulated. The heart stops, cardiac arrest, and the person dies. So this is euthanasia, and it's a strategy, and it has to be initiated by a patient. But the idea here is to show you how important the potassium ions are for maintaining the resting membrane potential. Okay, so now we move on to the local and graded potentials. So remember, the local potentials happen here at the dendrites and the cell body, and the action potential goes down the axon. So here are the channels, again, leakage and the sodium potassium. This is for resting membrane potential. Now we're going to focus here, and you're going to go into great detail and see the complexity of these gated ion channels. So these ion channels open upon stimulation, very different from these. Okay, so let's go now to the local action potentials, the local and the action potentials, and the gated channels. So there are different types of gated channels. Gated, that means they have a gate, they're closed. Upon a stimulus, they open. But what is that stimulus? The stimulus can be very different. You have some channels are chemically regulated. That means that there's, for example, a neurotransmitter that binds to the channel. So the channel is also a receptor. So the neurotransmitter binds to the channel and the channel opens. So this would be chemically regulated. It's actually a bona fide chemical, can be a neurotransmitter. It can even be an amino acid, because right now you only heard about two neurotransmitters, right? Acetylcholine and norepinephrine. But there are amino acids that are neurotransmitters also. Okay, so chemicals are released, they bind to the channel opens, so it's chemically regulated. Other channels are mechanically regulated. So the channels for sound receptors and so on, it's the pressure on the tympanic membrane that exerts pressure, and on the hair cells, the hearing receptors. So when there's pressure on the plasma membrane, the channel will open. So this is mechanically regulated ion channel. So this is a new concept that you learned today. So you have chemically regulated, but you also have mechanical regulated. And that's involved, for example, touch and hearing and so on. Anything that exerts pressure on the plasma membrane will open the channel mechanically. So these two, chemically regulated and mechanical regulated, these channels are involved in local potentials. The voltage-gated ion channels, those are always involved in the action potential. So here you see three types: chemically regulated, mechanically regulated, and voltage regulated. These two are involved in local potential. This one is involved in action potential. And remember that we have potassium-gated ion channels and we have sodium-gated ion channels. Is this what is meant by ligand-gated? The ligand-gated is here, the chemically regulated. A ligand is a chemical, synonymous. Ligand equals chemical. Do they all need ATP? This process of opening and closing the channels is not dependent on ATP, but there are other factors and other steps that are involved. Because remember here, the ATP is involved in the active transport when you're transporting the ions in the opposite direction. Here, for example, the sodium is coming from high to low, right? And the potassium is going from low to high. So they're not going against the concentration gradient. So in that manner, they don't need ATP. Okay, so let's now focus on the graded potentials. So an important property of the graded potentials is that they vary in amplitude. What does that mean? That means that they can go from minus 70 to minus 60 to minus 62 to minus 65 and so on, never reaching threshold, but they can vary. They're localized. That means they only travel a few micrometers. They only go through the dendrites or the cell bodies. They never make it to the axon. They travel short distance communication. So here's a chemically regulated ion channel. Now we're going to make things more complex so that they're more specific. Remember that all this is very important. All this knowledge is very important because it allows us to define different disorders, different diseases that might be affected by some type of channel and not other, and to design drugs that are specific for some channels and not others, right? So here are the chemical regulated or ligand regulated, synonymous, chemically or ligand. So these open in response to neurotransmitters or hormones. Okay, but there are two types of chemically regulated ion channels. Some open directly, so the channel and the receptor are the same. So that means the chemical binds to the channel and the channel opens. But others, the channel and the receptor are two different proteins. They're two different complexes. So that means the chemical binds to a receptor, and then once it binds to that receptor, there's a message that is conveyed to the channel. So that means that the receptor and the channel are two different entities, two different complexes. So let's look at this into more detail. So let's say up here, this is what we call an ionotropic. Tropic means change. So ionotropic, that means that the receptor for the chemical and the channel are the same complex. So the acetylcholine receptor here, when acetylcholine binds to this channel, which is a receptor in a way also, so when acetylcholine binds to this, the channel opens directly. You don't need any other step. So the channel and the receptor are the same. Now down here, this is a metabotropic receptor. That means you see that you have the receptor and you have the channel. And there's a difference here. It can be 10 to 200 nanometers. So that means they're on the plasma membrane next to each other, but they are different entities. So this receptor is also for acetylcholine. But in this case, it's known as a metabotropic receptor. So you see here, this protein spans the plasma membrane 7 times. So that means acetylcholine binds to the receptor. It causes a change in conformation. And then there are other proteins that are bound to the inside of this protein. And they come off and send a signal down the plasma membrane to the channel. And this would be a potassium channel. So that means that the receptor that receives the message from the ligand alters its configuration, its conformation, and sends a message to the channel, to the potassium channel. So this would be a metabotropic receptor. So you have ionotropic and metabotropic receptors, so a little bit more of complexity. So the only difference is that the indirect uses a messenger molecule, right? It uses a messenger molecule, and that message is generated by the ligand binding to a receptor. And then the message is conveyed down the channel. Exactly. Okay, so here you see again, I don't want to repeat, you see the ionotropic receptor. The ionotropic channel opens directly when the ligand binds to it. Here you have two different complexes. You have the receptor that responds to the chemical, to the neurotransmitter, and then a message is conveyed to the channel. So this is a metabotropic receptor. Okay, now let's move on to the mechanically regulated. So no chemicals now. Forget about chemicals. This is a physical action on the plasma membrane. So pressure is exerted on the plasma membrane. And you see here the channel is closed, the gate is closed. And you see here's a cytoskeleton. So you can have tubulin fibers or actin fibers. And when this pressure exerted on the plasma membrane, it pulls apart and opens the channel. So this is a mechanically regulated ion channel. Here it's closed, there's no pressure. When there's pressure on the plasma membrane, physical pressure by touching the skin or the sound, all these make pressure on the plasma membrane, will resulting in the opening of the channel. So you see here, applied pressure on the plasma membrane will open the gate of the mechanically regulated channel. And this responds to sound waves and touch. So it can be sodium, potassium, calcium, all can go in and out through mechanically regulated channels. Okay, so the graded potentials are localized in the dendrites and cell bodies. They vary in amplitude. They travel a few micrometers and they involve in short distance communication. Applied pressure by what? By, for example, your hand, right? Your hand on the skin is applying pressure on the receptors. Merkel, remember, Meissner, Pacinian receptors. So this pressure, physical pressure from your hands, an object can exert physical pressure. The sound waves is the way the sound generates waves, and the waves exert pressure. So it's physical pressure applied on the plasma membrane. Okay, so let's see here the graded potential. So here's the resting membrane potential. Then let's say there's a stimulus and the potential goes from minus 70 to minus 65. The channel opens. So that means sodium goes in. So it goes from minus 70 to minus 65 millivolts. Maybe this another stimulus comes along and it goes to minus 60 millivolts, but then nothing happens. That potential dies out. It doesn't travel in distance. So remember, I put here the car to remind me. Suppose you're in a car, you turn on the motor, and you just slightly press the accelerator. The car is not moving, but you're still generating some acceleration, but it's not strong enough for the car to move. So you press a little bit, and then you take out your foot, the car goes nowhere, although you're generating a little bit of acceleration. So it's the same thing. You have to press strong enough on the accelerator for the car to move. So the sodium diffuses from one area to another and can depolarize adjacent cell membrane sections. We'll come back to this. Because if you think, how does it go from minus 65 to minus 60? So because you have sodium here that is traveling by diffusion inside and changes the potential. Okay, now another layer of complexity, and that is the type of chemicals and the type of receptors that can be excitatory or inhibitory. So some receptors and some neurotransmitters can be the accelerator, can increase the acceleration in the car, but others can be the brakes, they can be inhibitory. They prevent the generation of an action potential. So you have ligands that bind to the channels and allow sodium in, potassium out. They can be excitatory. Also mechanical stimuli that open mechanical channels can be excitatory. And that's, in this case, you have sodium and calcium coming in. So they are excitatory. That means they go in terms of voltage, they would go from minus 70 to minus 65 or to minus 60. That will be excitatory. Why? Because it's going towards the threshold value, right? Excitatory. But they can be inhibitory. So let's say you have a channel that opens with a neurotransmitter that's glycine. So glycine is an amino acid that can be a neurotransmitter. And it is the inhibitory neurotransmitter in the spinal cord. In the brain, the inhibitory neurotransmitter is called GABA. Okay, so when glycine binds to this channel, so let's say glycine here is exerting the same function as acetylcholine, so it's a neurotransmitter. So glycine is released, binds to the channel. This channel now is a chloride channel. So chloride comes in, and now it becomes more negative. So that means that instead of being minus 70, now it's going to be minus 80. So that means it's inhibitory. It makes it more negative. So therefore, it's much harder to induce depolarization. So you can be excitatory, going towards threshold, or it can be inhibitory, going away from threshold. So going even more negative than the resting membrane potential. And you need both actions. You need both excitatory and inhibitory actions so that everything is going to be smooth. Okay, so let's see this in a graphical manner. So this graph, the y-axis represents millivolts, so the potential. The x-axis represents time in milliseconds. Okay, so minus 70 here is the resting membrane potential. Minus 80 would be what we consider a hyperpolarization. Hyperpolarization, because it's going below the resting membrane potential. When it goes to 60, that means it's being depolarized. It's going towards threshold. Threshold is here, minus 55 millivolts. So if you have a stimulus that causes an EPSP, what does it mean, EPSP? It's excitatory postsynaptic potential. So excitatory, that means it goes from, remember, we're talking about graded potentials that occur in dendrites and cell bodies. So you're going from minus 70, now you have a stimulus. It goes from minus 70 to minus 60, and then it stops, it dies out, and it repolarizes. It goes back to the resting membrane potential. So when you go towards threshold, that is depolarization. When you go back to resting membrane potential, that's repolarization. So here, this stimulus generated an EPSP, an excitatory postsynaptic potential. In this case, the stimulus, another stimulus, generates an IPSP, an inhibitory postsynaptic potential. And this all depends on which neurotransmitter is going to be released, right? If it's a neurotransmitter that opens a chloride channel, that's going to be inhibitory. If it's a neurotransmitter that opens a sodium channel, that's going to be excitatory. It's going to induce depolarization. So if you have glycine being released in the chloride channel opening, chloride goes in and makes it even, the postsynaptic neuron is going to be negative, more negative. So hyperpolarization. Depolarization is caused by sodium coming in, exactly. And hyperpolarization is caused by potassium going out. More potassium going out. Then it makes it more negative, right? So hyperpolarization involves potassium. And repolarization also involves potassium. So depolarization, you go from -70 to -60, that's excitatory. Hyperpolarization, you go from -70 to more negative, -90, it's inhibitory. Okay, so here we are. We discussed the gated ion channels that are chemically regulated or mechanically regulated. And these are involved in local potentials, and they can generate an EPSP or an IPSP, an excitatory or an inhibitory postsynaptic potential. So for hyperpolarization, does glycine always play a role in making the millivolts more negative? Yes, because glycine is an inhibitory neurotransmitter. Okay. Is repolarization and hyperpolarization the same? No. They're caused by potassium, but repolarization is when it goes from being depolarized to going back to resting membrane potential. That's repolarization. Hyperpolarization is when it goes below the resting membrane potential. What you mean you're cutting out like crazy? Oh, maybe it's your computer. Everybody doesn't hear me in a
No, I can hear you. I can hear you straight.
Okay. Okay. So enough about this. Now, just envision a postsynaptic neuron. Remember that I showed you that slide here, right? This is the pink is the postsynaptic neuron. And now you have all these axon terminals, and some are going to generate an IPSP, others are going to generate an EPSP. And it's the sum of all of those. When you add those all together, it has to reach -55 to go down to be converted to an action potential. Okay, so action potential. This occurs in axons. They travel a long distance and they depend solely on the voltage-gated ion channels. So here you see the different types of channels that we have been talking about. Here in the resting potential, we have the leakage channels, which are always open, they open and close randomly. Here are the graded ion channels, ligand gated or chemically gated, and the mechanically graded. So these are involved in graded potentials. Now here are the voltage gated ion channels. That means that the channels open when there's a change in voltage on the plasma membrane, at the plasma membrane. Okay, so here you see the outside, the channel is closed. You have positive outside, negative outside. Now, if there's a change in voltage that is originated by the graded potential, right? So if there's a change in voltage, now the channel opens. When it reaches threshold, then it becomes negative on the outside, positive on the inside, and the channel opens. This happens when the change in potential reaches threshold, minus 55 millivolts. So let's see here, resting membrane potential. This would be a voltage-gated sodium channel. If you remember, it has two gates, an activation gate and an inactivation gate. So question, are the leakage channels always open, or do they open and close randomly? They open and close randomly. Yes, this should be randomly, not always. Should be randomly. You are absolutely correct. Okay, so here we have the resting membrane potential scenario. Minus 70 millivolts. And you see how the voltage is measured. Is there a difference between threshold and subthreshold? Yes, subthreshold is 65 millivolts. That will be subthreshold. Okay, so now you have the stimulus that is strong enough and changes the potential at the membrane to threshold to minus 55 millivolts. Oh, so first let me focus here. So you have the activation gate closed, the inactivation gate open. This is a sodium voltage-gated ion channel. So now you have the threshold reaching minus 55 millivolts. The activation gate opens. So remember, these are all proteins, and they change in conformation depending, in this case, on the voltage. So when it reaches minus 55 millivolts, the gate opens, sodium rushes in, and it goes up to plus 30 millivolts. So from minus 55 to 30, this is the depolarization stage. Depolarization, going from minus 55 to plus 30 millivolts. So why doesn't it go to plus 40 or plus 50? Why? Because the inactivation gate closes at plus 30 millivolts. That means no more sodium can go in. So that means here, then there's no more excitability, because all the sodium channels will be closed. No more sodium can come in, and therefore you cannot excite this neuron anymore once all the sodium channels are inactivated. Okay, so this is the sequence. Minus 70 to minus 55. Then you go from minus 55 to 30. This is the depolarization state. Now, what happens after this? So, depolarization is very quick. That means sodium goes in, allows to go from minus 55 to plus 30. Now, from plus 30, it has to go back down to the resting membrane potential, and that is the repolarization stage, which represents the opening of the potassium channels. So potassium comes out, making the inside now going back to resting membrane potential. So depolarization involves the influx of sodium, and repolarization, the efflux, the efflux of potassium. So here are the different steps. Resting, you have sodium and potassium channels. They're both closed at resting membrane potential. Then there's a stimulus strong enough, and watch the curve on the graph. So there's depolarization reaching minus 55 millivolts. The activation gate opens and sodium rushes in. The potassium channel is still closed. Then when it reaches plus 30 millivolts, the potassium channel starts opening, inactivation gate closes, and potassium starts leaking, leading to repolarization. And then, because the potassium channels open slower than the sodium, then this goes or close slower than the sodium. You have a little bit of hyperpolarization going to minus 90 millivolts. And then the astrocytes get rid of the potassium and it goes back to resting. So resting, depolarization, repolarization, hyperpolarization, going back to resting. So the voltage-gated ion channels open when the membrane potential is minus 55 or above. Yes, that's the threshold. Okay, and you see the graph here. This is the local potential because it hasn't reached the minus 55, so it could die out. Once it reaches the minus 55, it goes up to plus 30. Then you see the potassium channels opening, starts to go down, the hyperpolarization, and then go back to resting. So you see here in a action, the change in potential in action. What I want to show here is the different stages of the opening and closing of the channels. So in the depolarization, you have the opening of the sodium channels. And around here, the potassium channels start opening. Here, the sodium channels are inactive. No more sodium can go in. And the potassium starts leaving for repolarization. Okay, so now we know what happens here, but how is it that this now is transmitted, is propagated down the axon? So you see here the different stages, the resting, the depolarization, and repolarization. But now here, the depolarization, you get a lot of sodium coming in, which leads to a change in the neighboring section of the axon. So it leads to depolarization of the axon neighborhood. Here, all I wanted to show is the generator potential, is the potential that induces all this mechanism of leading to an action potential. Okay, I think, let me see, 7:20. So just for a few minutes, I want to show here that there's going to be a stage where the neuron can no longer respond to stimulus, and that's the refractory period. Remember, we talked about the refractory period in the muscle fibers, where they could no longer respond to a stimulus. This same event happens in neurons. When all the sodium channels are inactivated, then the neuron cannot be stimulated, is no longer excitable. So that's the refractory period. When some of the sodium channels now go back to being like when they are at the resting membrane potential, then you have a super threshold stimulus that can open some channels, and that's known as the refractory period. So the refractory period is the time at which some of the sodium channels now recuperated. That means that the inactivation gate is open and the activation gate is closed so that they can be excitable. So that's the relative refractory period. But the absolute refractory period means that all sodium channels are inactivated. Okay, and why is this so important? This is important because it allows the action potential to travel just in one direction. So the action potential is traveling down the axon because you have waves of depolarization. But why can't it go back It cannot go back because of the refractory period. So this, it's going down, that traveling down the axon because the sodium is diffusing along the axon and therefore it's causing depolarization. But then it cannot go back because this area is is absolutely refractory. The the channels are all inactive. So that means the depolarization cannot occur backwards. It can only occur in one direction. And we'll go over this in the next class. We'll end here. Ooh, this is very intense, I would say. But I hope you understand all this and the importance of the ions and the channels and the properties of the plasma membrane. So we're done. Any questions? Okay. So see you Thursday and good luck on your quizzes. Bye bye.